So today I'm going to be talking about the Mendel's genetics and the laws of inheritance. So if you're watching this video, you probably know a decent bit about Mendel's experiments. So I'm just going to go over them really briefly. So his experimental aims, he wanted to discover the link between the phenotype of parent and their offspring. So before Mendel's time, they just thought that the parents will have an offspring and it will just be a mix of the parents, and that's how genetics, like the diversity, occurs. But he thought, how does this make sense? There'd be no genetical variance, as it just keep being a mix of the parents to the offspring. He believed that it was not just a mixing of characteristics. So he used Bacillum stativum, which are a type of peas. And the main reason he used these peas were because they had a short generation time, so he could keep making the offspring quickly. Many offspring produce quickly, so you didn't have to wait. Cheap and easy to grow. This is the main point, because you don't want to use up too much space. You don't want to use up too many resources either. They used to self and cross-fertilize. These 2 points are very important. We'll come back to them later. They produce discrete traits, so you could able to compare just with your eyes visibly. And they all are encoded by a single gene, all the different traits. Now, he didn't know this at the time, but it's almost, I wouldn't say luck, but it's very important that these P's that he used were all encoded, all the different characteristics were encoded by a single gene. This allowed his experiments to be, this basically allowed his experiments to work pretty much. The traits he looked at, so from these P's, he looked at 7 particular traits and he compared them. He looked at pod color, so yellow or green, pod shape, crinkled or quite flat, seed color, yellow or green, seed shape, again wrinkled or smooth, flower color, flower position, stem length, and again these are all encoded by a single gene. How Mendel carried out his experiments. So first he wanted to make sure a true homozygous breeder of a specific phenotype so he made breeding lines. So for example, if he wanted to get only yellow plants, he would self-fertilize these, only wanted yellow peas, he would self-fertilize these yellow peas continuously until he only was getting yellow peas, to make sure that these were homozygous. Next, he conducted a monohybrid cross. So this is when two homozygous alleles are crossed together. So how he did this, he brushed pollen from one homozygous breeding line onto another with a different phenotype. So for example, if he mixed green peas and yellow peas, they would be mixed together, and he would then cut off the stamens for self-fertilization, because he'd want to make sure that the two that he was controlling would specifically be in contact with each other. This would perform a cross, so they would fertilize and the gametes would cross over. The alleles would cross over. And he performed paired crosses with different versions of the same characteristics, and then observed the F1 generation. F stands for FIFO. So this is what his first generation will look like, with large G being green peas and small g being yellow peas. So he'll cross over 2 homozygous colors of pea. So the G, large G signifying green and the small signifying yellow. So as you can see, when he crossed them over, he would get all green peas, as large G would be the dominant allele. However, this is where it got interesting. In the second generation, although the first generation he was using were all green, it was the first time he discovered that it would give out a yellow pea. So 2 green peas would be crossed, and it would give out yellow pea with 2 small g's. So you're getting GG, which is homozygous, big G, little g, and another big G, little g, which are both heterozygous and a homozygous little g. And this was the first time that anyone had fully proven that parents, that genetics isn't just a mix of your two parents. This was the famous example of his 3-to-1 ratio. So now we're going to go over the two laws that he found. They're very simple. The first one is Mendel's first law. This is also known as the law of segregation. The two is basically the two different alleles of any individual will pass into different gametes. So if you have, for example, the same with the peas, if big G is green and little g is yellow and big G is dominant, they'll split into different gametes with the large G going into one and a small g or recessive yellow going into the other. Mendel's second law is also known as the law of independent assortment. This is basically all the alleles are randomly assorted and are passed on to offspring with an equal probability. Now, this may seem obvious, but at the time it wasn't. The fact that different, for example, we'll go back to the yellow and green peas, that a small g is much as likely to be passed on as a big G. Thank you very much for listening.